Early developmental processes and overview. Cleavage. After fertilization, the development of a multicellular organism proceeds through a process called cleavage, a series of mitotic divisions whereby the enormous volume of egg cytoplasm is divided into numerous small nucleated cells. These cleavage stage cells are called blastomas without increasing cytoplasmic volume by abolishing gap periods G1 and G2 phases of cell cycle. So, rate of cell division is controlled by the proteins and mRNA stored in the oocyte. Fertilization activates MPF, mitosis performing factor, responsible for resumption of mitotic cell division in the ovulated frog egg. Then blastomas generally progress through a biphasic cell cycle consisting of two steps, M and S phase. The cell cycle is independent of nuclear genome for a number of divisions, which is rapid and synchronous. As the cytoplasmic components are used up, the nucleus begins to synthesize new cytoplasmic components by zygotic gene expression. The embryo now enters a mid-blastular transition in which several new properties are added to the biphasic cell divisions of the embryo. The gap stage G1 and G2 are added to the cell cycle. The synchronicity of cell division is lost because different cells synthesize different regulators of MRF. New mRNAs are transcribed, encode a protein that will become necessary for gastrulation. Early blastomas, normal cells are there. So the cytoskeleton mechanism of mitosis. Cleavage is the result of karyokinesis and cytokinesis. The agent of karyokinesis, mitotic division of cells nucleus, is the mitotic spindle with its microtubule composed of tubulin proteins. The agent of cytokinesis is a contractile ring of microfilaments made of actin. The mitotic spindle and contractile ring are perpendicular to each other and the spindle internal to the contractile ring. The contractile ring creates a cleavage furrow, which eventually bisects the plane of mitosis, thereby creating two genetically equivalent blastomas. The placement of centrioles is critical in orienting the mitotic spindle and thus the division plane of the blastomas. And the blastoma is separated into dorsal or ventral daughter cells, anterior and posterior daughter cells, or left and right daughter cells. Then pattern of embryonic cleavage. The pattern of embryonic cleavage is determined by two major parameters, the amount and the distribution of yolk protein within the cytoplasm. Factors in the egg cytoplasm that influence the angle of mitotic spindle and the timing of its formation. Then yolk inhibit cleavage. The yolk-rich pole is referred as vegetal pole and the yolk concentration in animal pole is very low. Eggs that have sparse and equally distributed yolk are known as isolecithal eggs, and these species cleavage is holoblastic. Cleavage furrow extends to the entire egg. Zygote containing large accumulations of yolk undergo meroblastic cleavage, where only a portion of the cytoplasm is cleaved. Then protostome, a multicellular organism whose mouth developed from a primary embryonic opening, shows spiral cleavage, annelids, mollusks, flatworms. Then dipterostome, a multicellular organism whose anus developed from a primary embryonic opening, shows radical cleavage, echinoderm. 8 egg cell stage, spiral cleavage, gastrulation, archendron, coelom, mesoderm, blastopore, mouth, protostomes, anus develops, deuterostomes, mouth. Does the blastopore become mouth in case of protostomes? Okay. In case of deuterostomes, blastopore becomes anus. Okay. Schizocoelus versus Enterocoelus. Schizocoelus, the condition of embryonic development in which the body cavity is formed by the splitting of the mesoderm. Coelom begins as splits within the solid mesodermal mass. Cleavage holoblastic, spiral and determinate. Occurrence protostomes. Examples annelids, arthropods and mollusks. Then Enterocoelus, the condition in which the coelom forms from pouches pinched off of the digestive tract. Mesoderm arises as lateral outpocketing of archendron with hollows that form coelomic cavity. Cleavage radial and intermediate. Occurrence deuterostomes. Example echinoderms, hemichordates and chordates. Then gastrulation and morphogenetic movement. By the end of cleavage, the blastomas have usually formed a sphere known as a blastula. After the mitotic division slow down, the blastomeres undergo movements and changes that relate their positions to each other. The series of cell arrangements is called gastrulation, and the embryo is said to be gastrula. As a result of gastrulation, the embryos contain three germ layers that will interact to generate the organs of the body, organogenesis. The patterns of gastrulation vary enormously throughout the animal kingdom. There are only a few basic types of cell movements as follows. Imagination, the folding of a region of cell. So the patterns of gastrulation vary enormously throughout the animal kingdom. There are only a few basic types of cell movements as are follows. Invagination, the infolding of a region of cells. Involution, the inward movement of an expanding outer layer that is spread over the internal surface. Imagination, the infolding of region of cells. Involution, the inward movement of an expanding outer layer that spreads over the internal surface. Then ingression, a migration of the individual cells from the surface layers into the interior of the embryo in the form of mesenchymal cells. Then delamination, the splitting of one cellular sheet into two or more or less parallel sheets. Then epiboly, the movement of epithelial sheets that spreads as a unit to enclose the deeper layers of embryo. Imagination, infolding of cells sheet into embryo. Example, sea urchin, endoderm. Involution, turning of cell sheet over the basal surface of an outer layer. Ingression, migration of individual cells into the embryo. Sea urchin, mesoderm, Drosophila, neuroblasts. Then delamination, splitting or migration of one sheet into two sheets. Delamination, mammalian and bird hypoblast formation. An epiboly, the expansion of one cell sheet over other cells. And ectoderm formation in amphibians, sea urchins, and tunicates. Types of cell movement during gastrulation. Cell specification and axis formation. Cell fates are specified by cells-to-cell signaling or by the asymmetric distribution of patterning molecules into particular cells. Such asymmetric distribution of patterning molecules begin during cleavage and passes through the egg cytoskeleton. So embryo must develop three crucial axes that are the foundation of the body: the anterior-posterior axis, the dorsal-ventral axis, and the right-left axis. Anterior-posterior axis is the line extending from head to tail, and the dorsal-ventral axis is the line extending from back to belly. Then the right-left axis is the line between the two lateral sides of the body: dorsal, ventral, anterior, posterior, lateral, horizontal plane, and mid-sagittal plane. Dorsal, ventral, lateral, horizontal. Early development in sea urchins. Sea urchins exhibit radial holoblastic cleavage. First and second cleavage both are meridional and are perpendicular to each other. The first cleavage is equatorial, perpendicular to the first two cleavage planes. After third cleavage, the four cells of the animal tire. Next, we will be learning about early development in sea urchins. Sea urchin exhibit radial holoblastic cleavage, we just mentioned. First and second cleavage both are meridional and are perpendicular to each other. Okay. The third cleavage is equatorial, perpendicular to the first two cleavage planes. After third cleavage, the four cells of the animal tire divide meridionally into 8 blastomeres with the same volume. These 8 cells are called meromeres. The vegetal tire, however, undergo unequal equatorial cleavage, produce 4 large cells, the macromeres, and 4 smaller micromeres at the vegetal pole. Animal pole, vegetal pole. Animal half, vegetal half, meromeres, mesomeres, micromeres, and macromeres. Okay. Then fate map and the determination of sea urchin blastomeres. Animal pole, vegetal pole, oral, aboral. Large micromere, small micromere. The large micromeres become autonomously specified by maternally inherited determinants that had been deposited at the vegetal pole of the egg. And the cells are determined to become skeletogenic mesenchyme cells that ingress into the blastocoel and become skeleton of the larva. The large micromia produce paracrine and extracrine factors that simply specify the fates of their neighbor cells. Micromia transplanted from vegetal pole into the animal pole of host embryo. Transplanted micromia differentiate into skeletogenic mesenchyme and induce animal cap cells to become endoderm cell and form a secondary archendra. The initial regulatory transcription factors are disHAVed and beta-catenin, both of which are found in the maternal cytoplasm and more inherited to the micromeres. During oogenesis, disHAVed become located in the vegetal cortex of the eggs, where it prevents the degradation of the beta-catenin in the micromere and macromere cell. The beta-catenin is therefore allowed to enter into the nucleus, where it combines with the TCF transcription factor to activate gene expression. During normal sea urchin development, beta-catenin accumulates in the nuclei of vegetal cells fated to become endoderm and mesoderm. In embryo treated with lithium chloride, beta-catenin accumulates in the nuclei of all blastula cells by LICL blocking the GSK3 enzyme of the WNT pathway analog to disHAVed and transforms the presumptive ectoderm into endoderm, and animal cells become specialized as endoderm and mesoderm. When beta-catenin is prevented from entering the nuclei, it remains in the cytoplasm. Vegetal cells' fates are not specified, and the entire embryo develops as a ciliated ectodermal ball. Then specification of the vegetal cells. Maternal beta-catenin and OTX activate the BLIMP1 gene, whose product activate a gene encoding WNT8 that activate the genes for beta-catenin in autocrine manner. The juxtacrine signal delta protein formed from micromia activating the notch protein on the adjacent cells that tells cells how below them to become the non-skeletogenic mesenchymal cells. An axis specification. In sea urchin blastula, the animal vegetal cell axis appears to structure the future anterior-posterior axis, with vegetal region sequestered those maternal components necessary for posterior development. The oral laboral axis appears to form through the activation of the nodal TGFB genes in the oral ectoderm. Then gastrulation, ingression of the skeletogenic mesenchyme, invagination of the archendron. Early development in tunicates. Ascidians are characterized by bacterial bilateral holoblastic cleavage. Autonomous specification is seen in tunicate gut endoderm, muscle mesoderm, skin ectoderm, and neural cord. So conditional specification is seen in the formation of the brain, notochord, heart, and mesenchymal cells. Macho-1 mRNA was concentrated in the vegetal hemisphere of the unfertilized egg, but by the 8-celled stage, Macho-1 mRNA is found only in the B4. 1. Plastomeres that give rise to the muscle cells. Macho-1 activates TFS cascade that promote muscle differentiation while at the same time inhibiting notochord specification by inhibiting brachyury. Then autonomous specification of the endoderm originates from vegetal A4-1 and B4.1 blastomeres through the accumulation of B-catenin. Conditional specification of the mesenchyme and notochord by the endoderm. Tunicate gastrulation is characterized by the invagination of the endoderm, the involution of mesoderm, and epiboly of the ectoderm.